All right, for this explainer, we are going deep. We're getting right down to the cellular level of the asthmatic airway. We'll be breaking down a really critical review on mucus hypersecretion to understand exactly what's driving it, from the cells that make the mucus to the very genes that code for it. So here's the game plan. We're going to start with the main structural change you see in the airway. Then we'll dig into the genetics and the signaling pathways that are responsible for it. And then we're going to uncover a clinical paradox that honestly complicates the whole picture. Okay, let's kick things off with the absolute core structural abnormality in an asthmatic airway, this huge increase in the number of mucus-producing goblet cells. It's a phenomenon known as goblet cell hyperplasia, or GCH for short. And look, this isn't just some vague observation. Using a really rigorous method called design-based stereology to quantify 3D structures, researchers found that people with asthma have, on average, 2.5 times more goblet cells in their airway lining than healthy people. That is a massive increase. So what's the upshot of having all these extra cells? Well, it's pretty straightforward. You get a dramatic increase in stored mucin. The total volume of this stuff just sitting inside the epithelium is about 3 times higher in asthmatics. That creates this huge reservoir of mucus, just waiting to cause airway obstruction. Okay, so we've seen the cellular change. Now let's peel back another layer and see how this is all programmed at the genetic level. We're going to look at the expression of the muc genes. These are the genes that provide the protein backbone for all that mucin. Now what's really fascinating here is that this isn't just a simple across-the-board ramp-up of all mucins. Not at all. Instead, asthma has this very distinct genetic signature. MUC5AC, which is the dominant gel-forming mucin in everybody, shows way higher expression in asthma. So the really critical point here isn't just more mucin. It's this very specific shift in the mucin profile. Yeah, MUC5AC is the main player, but you also see the significant jump in MUC2 and MUC4, and get this, a significant drop in MUC5B. This altered recipe of proteins almost certainly changes the physical properties of the mucus itself, affecting its stickiness and how easily it can be cleared. So the big question is, what's pulling the strings here? What's controlling the expression of these MUC genes? Well, tons of evidence from animal models points to one specific arm of our immune system, the T helper type 2 cells and their chemical messengers, the cytokines. And the evidence is, well, it's pretty overwhelming. If you overexpress Th2 cytokines, we're talking about molecules like interleukin-4, IL-9, and IL-13, in animal models, that alone is enough to cause goblet cell hyperplasia. These molecules are the primary drivers, signaling for both more goblet cells to grow and for other cells to turn into goblet cells. This right here kind of lays out the domino effect that's been proposed. Cytokines like IL-9 and IL-13, they don't act in a vacuum. They actually leverage other signaling pathways. They fire up the epidermal growth factor receptor system, and they induce these key ion channels. All of these signals then converge on ramping up MUC5AC expression, which seems to be the critical trigger that makes an ordinary epithelial cell decide to become a goblet cell. All right, let's zoom in on one of the most surprising parts of this whole pathway, the role of ion channels. I mean, you don't usually think of them as drivers of cell differentiation, right? But it turns out they're absolutely critical. First up, we've got HClCa1. This is a calcium-activated chloride channel, and it gets switched on directly by those Th2 cytokines. And here's the remarkable part. If you just overexpress this one single channel in airway cells in a lab, that alone is enough to make them transform into mucus-producing cells. It's an incredibly powerful downstream effector of that Th2 signal. And the second key player is NKCC1, a sodium-potassium chloride cotransporter. This channel is found to be specifically ramped up in the fully formed goblet cells of human asthmatics. So this suggests a kind of division of labor. HClCa1 might kick off the transformation, but NKCC1 could be crucial for actually maintaining the cells and their ability to secrete all that mucus. Okay, so let's recap. We've got an airway that is just loaded for bear, right? 2.5 times more goblet cells, holding 3 times the normal amount of mucin. You'd think that the moment an asthmatic gets exposed to an allergen, boom, all that mucus gets released. But let's see what the clinical data actually shows. But first, just a quick look at how this secretion process works, because it's pretty incredible. Inside the cell, mucin is packed in super tight, all condensed by calcium. But when it's time to secrete, the granule merges with the cell surface. Calcium flows out. and water rushes in. This allows the mucin molecule to just explode outwards, expanding rapidly like a jack-in-the-box. So this explosive mechanism really sets up the central question for us. With this massive system, preloaded and ready to go, what happens during a real-life allergen challenge in a human subject? Do they pull the trigger? And the results were just not what anyone expected at all. The allergen challenge definitely provoked a powerful inflammatory response. You can see eosinophil numbers shoot up in the tissue just an hour later. But amazingly, the volume of stored mucin did not go down. And this graph just drives that paradox home. Not only was there no degranulation at 1 hour or even 24 hours later, there was actually a tendency for the amount of stored mucin to increase by the 24-hour mark. The inflammatory trigger was pulled, but the goblet cells, they just didn't fire. So what this tells us is that the link between that allergic inflammation and the actual release of mucus, it's not a straight line, not even close. The only logical conclusion is that there must be powerful inhibitory mechanisms actively preventing these loaded goblet cells from firing, even right in the middle of an allergic reaction. And that leaves us with this massive critical question. What are these protective brakes? And maybe even more importantly, what causes them to fail during a severe asthma attack, when mucus plugging can become deadly? The review suggests something like a viral infection on top of the allergy might be the second hit that's needed. Unraveling these inhibitory mechanisms is a huge frontier for developing new therapies that can stop that catastrophic mucus release.